What is tagging? Normally, every day in our daily life, you use the word tagging. What is tagging?
Compiling together or filing things together.
Tagging. Just tag means you are giving some name. You just tag someone. Like for example, how you tag your plants. You will assign some number or you will assign some address. Okay, it is this particular plant is in the particular row, is in the particular line. Means we are giving some address to it. Means the gene tagging in the same way. In the genome, for example, your flower color is there. The gene responsible for flower color we get to know. But where it is located, we are tagging the gene responsible for, we are tagging a gene for a particular trait and also where it is located in the genome, where it is located in the genome. This is what the problem. Admitting, so I will give a 2 minutes time to join. Please join within the time. Okay. Where it is located in the genome. We need to identify that. What are the two things we are doing? For example, you are doing a phenotyping. You want to assign particular gene for the trait and also along with that you have to assign where the gene is located in the genome. That is one of the uh, goal of a breeder. You are studying for a particular trait. Then other things what we will do in the molecular breeding for how you will do. I told you have to assign a gene for a particular trait and you have to tag in a particular genome. You have to give an address for the gene. How you can do with the knowledge of your linkage and recombination? We already discussed. You have to tell
Or
how you will do. What are the
by checking
possibilities?
by checking which marker is always associated with that particular trait. Like the marker will be there when the particular trait will be there. Likewise, is it
Exactly. Marker and the trait. But how you know that particular marker is associated with the gene? How you
will
as-
generate,
how you will associate the marker with the gene?
sir, we will generate some manipulations and see whether in all the recombinants that the marker is associated with the gene, sir.
Manipulations. I'm hearing first time. What manipulation you are going to do? Either you will talk with the genes.
No, sir, like we create some populations according to the trait and something we are
Exactly.
going to do with the recombination frequency, sir.
Yeah, not something. You should do one thing. Okay. First, we will cross.
Lots good, sir.
Yesterday we discussed. Last class we had discussed. We cross extreme parents of the particular trait. For example, flower curl, white and red. Both extreme parents. We will generate a mapping population. We will identify. We will genotype with the population. Today we are going to discuss. Then we will do the assays. Okay, means now you got to know what is gene tagging means. You are just assigning where you are, for a particular trait, which gene is responsible and where it is located and which markers are associated with them. This is the only the entire molecular breeding is revolves around it. This is the one of the central focus. Then how you can do it, we will discuss in detail. Why you? Okay, you told mapping the gene, tagging the gene. Why we need to map a particular gene? What is the need of mapping the gene? Actually, in ORW, like many people, many MSc students were presenting molecular mapping for some particular traits. In every crop, they are doing molecular map. So become a trend. The final question asked by reviewers are why you are mapping a gene? Why we need to map a gene? Discuss this. You should tell.
To see the location of
Okay,
particular trait.
we don't know the location. Then
Sir,
what we will do?
we only by knowing the markers which are linked to the gene in that particular chromosome in that location, we can do indirect selection or perform other tasks
Exactly.
easily without environmental
Exactly.
factors or
Indirect selection. In the first class, in the molecular application in plant breeding, we'll discuss why we need a markers. In that class, I have discussed in detail why we need to map a particular gene. We know that the phenotype is always associated with the genotype and environment. Discussed in first class itself. How the phenotype is affected by the genotype and the environment. The Johansson in his experiment is proved that phenotype is always a manifestation of the genotype plus environment. Another component is missing, G and E also. When particular genotype grows under multiple location, it having a different behavior. That is called a GE interaction. That also manipulates your phenotype. Means genotype, environment, G and E environment. But only heritable portion is genotype. Okay. But if your selection only based on the phenotype, the trait which are the effect is contributed by the environment and effect contributed by G interaction will not going to inherit for the next generation. You will not going to get any genetic gain in the next generation. We discussed in detail. That's why we need some method which indirectly selects the particular phenotype that is based on the genotype. Means for example, you take molecular markers. Molecular markers are means they are genotype. They are genes. Okay, they are genotype. They are present on the genome. We are selecting the genome. If you select the molecular markers means we are selecting the genome based. You are directly selecting the genotype. You are completely avoiding the environmental effect. Means P is equal to become G. Okay. Because your selection is based because the marker is, as I told, it's an independent of environment. Marker is an independent of environment. If your selection based on the marker, then your phenotype is exactly become the genotype. Then your selection will improve and your genetic gain will be improved. And we discussed in detail what is genetic gain, what are the components affecting the genetic gain. See, every class is interlinked in the plant breeding. Okay, please watch my recorded videos for before attending my next videos. Okay? Then, if you don't need to map a gene, if P is equal to come with G, means if the phenotype, for example, this is the phenotype, it's clearly visible. The color is clearly visible. You easily select the yellow color plant. But if it is a disease resistant, what you have to do for the selection? You have to do inoculation, we have to do all the things which I already discussed, which are all the things, then you can able to select. But it's affected by the environment, affected by the temperature, it's affected by your human errors. If you are not inoculated some plant, you are inoculated with higher inoculum load some plant, you are inoculated with lower inoculum load with some plants, then all the human errors, experimental errors, everything will mixed up and your selection will go wrong. Your selection will go wrong. That's why we need to map a gene and we need to identify the markers. Then we have to go for marker assisted selection. Why we need to map genes? We need to go for marker assisted selection. This is the one important point. If everybody asks you why we have to go for mapping a particular gene, the main objective of the breeder point of view, you should tell we can use them in a marker assisted selection. There are different schemes like marker assisted backcross breeding, marker assisted recurrent selection, genomic selection. Okay, n number of forward breeding with the markers. There is a number of marker assisted selections were there. We will discuss after discussing how to map a QTLR gene. Then, how we can construct the linkage map. We discussed in detail based on the recombination frequencies. 1% recombination frequencies is equal to 1 centimark. It's a started event in 1932 only he identified. He crossed some individual, he generated a population, then he identified how many number of recombinant individual, how many number of parental individuals, how many number like total recombination by total into 100 you will get a recombination frequencies. Then 1% recombination frequency is equal to 1 centimorg. But there are several exceptions for this. We will discuss. We already discussed Kosabi mapping, Halden mapping. So then, how using a marker, how we can construct a linkage map? First, you have to select two genetically divergent parents. For example, parent 1 and parent 2. Yesterday I told highly resistance crossed with highly susceptible. Means if the diverse, more will be the diverse, more will be the recombination, higher will be the resolution of your map. Then after crossing, you will generate a mapping population. We discussed what is different mapping populations, RIL, FEP2, Evacross, NILS. We'll generate a mapping population. Then after you select some marker systems, either SSR markers, either SNP markers, either any markers if you have available. Normally, generally we use SNSR and SNP. Take the marker and run in the parents. Then you will going to identify a polymorphic marker. The marker which is polymorphic between the parents, that marker is only used for construction of linkage mapping. For example, if the marker is showing a monomorphic band like this, like it is 50 base pair and 50 base pair here, means you should reject. But in parent one is showing 50 base pairs, but in parent is showing 60 base pairs. This is polymorphism. Means there is a different types of bands will be observed between the different parents. This is called as polymorphic. We discussed. Then after identification of polymorphic markers, you use, for example, you run 2000 SSR markers. Out of 2000 SSR marker, you are identifying only 200 polymorphic markers. Use them only. Use 200 polymorphic only. And none of the other 1800 marker which are getting monomorphic, you should reject them. After identifying the polymorphic marker, you have to genotype with all the individuals. First, you have to run all 2000 markers between the parents. Then you should identify the polymorphic markers. Then use polymorphic markers for genotyping of entire mapping population. For example, you have 300 mapping population. Use 200 markers to genotype 300 population. Like this, you can genotype all the individuals of the mapping population. Then all the individual population are screened with the polymorphic markers. Then after genotyping, I will show practically today how we can construct because I did. After that, then you will calculate the recombination frequencies and genetic distance. By genotyping, after genotyping, which one is polymorphic? After scoring, I showed, I think, how to score the individual parent 1 and parent 2. We will genotype and we will have to score and we have to create the genotypic data. Then by using genotypic data, we can construct the maps. Okay, marker 1, marker 2, marker 3, marker 4. How we can going to map? We will discuss in next slide. Then we had to do phenotyping of the entire mapping population, the genotyping of entire mapping population. Then use some software like marker or ICM mapping. There are QTL cartographer. There are any map maker. There are different softwares are available. You just run in them. Then you will get a good QTLs are genes associated with your trait of interest. This is the end. This is the only thing we will do in molecular breeding. There is another type. This is the by biparental linkage mapping. There is another method in association mapping also. We can map the gene, but as of now, we're just studying about linkage. Then,
So one
how do
doubt,
you
sir, from
phenotyping?
steps,
Yeah, yeah.
steps after genotyping the markers in all the mapping population individuals, we will
Yeah,
get
yeah,
the linkage map. Then only
yeah,
we
yeah.
are going to phenotype, sir. After only you
No,
are
no,
going
no.
to
It is
phenotype.
going hand in hand. In field, you do
Okay, okay,
phenotype. In lab,
okay,
your genotype. No worries.
okay, okay.
See, for example, you're flowering. You are working for the flowering trait, but don't wait up to flowering because DNA is same in the seedling stage, and the DNA is same at flowering stage. If you take the seeds, grow plants in a germinator, after 6 days from seedling, you extract the DNA. For example, you take rice will flower after 60 days, 30 days, like 40, 50 days. Okay, after that. Whole month you can do your genotyping. You finish genotyping before flowering. You can save the time. That is the one of the advantage of using markers. You can save the time of breeding. Okay. This is how you can save the time. Then phenotyping. How we can phenotype? There are different traits. Some are quantitative traits, some are qualitative traits, okay, which can be measured. But whatever the information you give to machine, it will give the genotypic data. Your interference in the genotypic data is nothing. You throw something, it will give something. But whenever you do mapping, phenotyping is very, very important. If you do wrong, like if phenotyping is wrong, even though genotyping is good, everything will mess up. For mapping, both phenotype and genotyping should be very good, and phenotyping is very, very should be very precise. After mapping a particular markers to gene, then by using only marker, we can do indirect selection. But for mapping, phenotyping is should be very, very accurate. If you do anything in the phenotyping, everything is going to mess up. Then you should be no missing data generally because if you are taking 300 individuals, you should have phenotypic data of all 300 individuals. If you miss any individuals, it will create errors. Then sample size. For example, you're having 3000 population. You can't able to screen all the 3000 with SSR. It takes a lot of time. Then you have to do sampling. That sample size should be very accurate. Then you should grow in a multi-location trials so that you can eliminate your QTL into environmental interaction. G inter interaction you should eliminate by growing in a multi-location trials. Then use phenotyping techniques like phenomics. Advanced phenotypic techniques means you're accurately phenotyping should be must by using phenomics or by using multi-year data, multi-location data. Okay. Then you should
Sir,
not miss any phenotypic data of individuals. It's how to phenotype the mapping population. You're asking something?
yes, sir, yes, sir. Sir,
Yeah.
even though we will normally sample the phenotypes, sir, in sample some individuals, or we will go entirely for the all the individuals, sir. Which
For example,
even
no, no, no. You have to go entire population. Genotyping, phenotyping of entire population. But sample size means, for example, you're having 3000 accessions. You had to select only few accessions. Okay. For example, rice, one plant, it will produce so many number of seeds, like nearly 1000 seeds you take. You can't use all 1000 individual for mapping purpose. You just select 200 or 300 individuals and then phenotype all 300 and genotype all 300. That sampling should be very nice. Okay, it should cover all the recombinant individual. Even though sampling is very less, but it should cover all the recombinant individuals. Okay, that is what a sampling.
yes, sir, understood, sir.
This is about phenotyping of the mapping. After phenotyping, you have to do genotyping. How to do genotyping? See, this is actually my slide only. This is some my seminar slide. For example, this is the ladder. First, you have to do polymorphic survey between the parents. See this marker, one band is here and another band actually it is here. To observe clearly, there is a band here, one band is here. Then in individual, this is mapping population like 1 to 21. In one gel, we can run only like in one lane, can run only 24 or 23, I think. Then in the second line, again 22, 23, 24, 25, 26, 27, like this, there will be 7. See, in parent 1, parent 2, in parent 1 band like this, in parent 2 band like this. In individual 1, we are getting band like this. In individual 2, band like this. Means you have to score. This is parent 1, this is parent 2. Here, this is parent 1, and this is parent 1, this is parent 1, this is parent 1. Sorry, this is below parent 2, this is below parent 2, this is below parent 2. See, this is heterozygous. Two bands are there. This is heterozygous between these parents. Then 11. This is parent 2, this is heterozygous, this is parent 2, this is parent 1, this is parent 2. Like this, you have to score the individual. Then you have to generate the data. Okay, marker 1, 0, 1, 2. For example, 0 is parent 1, 1 is parent 2, and your 2 is heterozygous. You have to give some codes like the 0, 1, individual 1, individual 2, individual 3, individual 4, individual 5, and marker. Then you have to generate genotypic data like this. You will generate one Excel sheet of scoring data. Then this will be called as your genotypic data. Then you have some Excel sheet of your traits like flower color and individual 1, individual 2, individual 3. For example, plant rate 10 meter, 12 meter, 13 meter, 14 meter plant. Then one Excel sheet of phenotypic data, one Excel sheet of genotypic data. You use both the Excel sheet and you will any use marker like a map maker, title cartographer. Then you will QTL will come. It's very easy. First,
Uh,
you have to develop the mapping population, genotyping of the mapping population. Then you have creating the linkage group based on the recombination frequencies. Then you should find the best possible order like marker 1 after marker 3 after marker 4, like order of the markers. Then your linkage map is constructed. Then use this map for the associate map with phenotype with some software. You will get a QTL or a G. It's a very, very simple procedure. But we'll discuss in detail how we can do that. Then how we can estimate the recombination frequencies. Number of recombinants divided by total number of individuals. This is normally direct count method which are earlier using for study. But many software they are using maximum likelihood method. Don't remember this formula, even I also not understood. Like it's a probability equation. Like it based on like the probability of the combination of occurring the recombinants with recombinants minus one minus recombinant. Okay, this is all probability, but normally we're using maximum likelihood method. It's entirely dependent statistics. Then normally we know that recombination frequencies are not exactly equal to your genetic distance after 20 centimorgan. Why it is so? We discussed because of double crossovers. To avoid that, we have to use some correction factors. That correction factors are Halden correction factor and another one is Kosambi correction factor. Halden correction factor, it will not include interference and it will correct like by using this formula. Then Kosambi, we already discussed in detail. I will not bore you by discussing again. Then another thing is logarithm of threshold. What is logarithm of threshold? It means it's the ratio between the likelihood of the genotype of the phenotype with linkage divided by the likelihood of the genotype or phenotype with no linkage. For example, you take log value of 10 means log value of 100, log value of log 1, log 2, log 3. Log 1, log 2, log 3. Log 1 means logarithm of threshold 1 means your 10 times the particular genotype or a marker is associated with the particular phenotype with the linkage than without the linkage. Means you can say that the 10 times the more probability is there, your particular marker is linked with your genotype, sorry, phenotype. This is one. Lord 2 means 100 times the probability of a marker is linked with your phenotype. Lord 3 means 1000 times the marker is linked with the genotype to the no linkage. Means if the Lord 1 means 10 times the probability, Lord 2 means 100 times the probability, and Lord 3 means 1000 times the probability, you can say that the particular marker is linked with the phenotype. Means this normally for if you have to say this marker is linked with the particular QTL or a gene, you should consider log 3 as a maximum types. Means if anything is crosses log 3 value, that any marker interval or a marker it crosses log 3 value, above the log 3 value, that marker is linked with your trait of interest. Understood what is log values?
Yes, sir, understood, sir, but one doubt, sir.
Yeah, yeah,
The software only calculate it, sir. Isn't it, sir?
yeah, we
One,
can't do,
one
we
time.
can't do anything, we
Yes, sir,
can't do
we
here.
can't do. Yes, sir, that's
This is highly, no, nothing. See, these are very easy thing, but you should upload the in software, it will calculate the lot. You are not going to do anything. Okay, just remember what is that. For example, lot 1 means the 10 times the 10 by 10 times you can say that the particular marker is associated with the linkage towards the 10 by 1. If lot 2 means 100 by 1, lot 3 means 1000 by 1. 1000 times with the linkage to the non-linkage. If lot 2 means 100 times to the linkage to the non-linkage, lot 1 means 10 times to the linkage to the non-linkage. Sorry, non-linkage. Normally, if you want to say some marker is associated with the trait, you should always consider above lot 3. Even nowadays, above lot 4, they are asking. Okay. If you want to say markers, if you want to say, okay, some QTL is there, means you should cross above lot 3 value. We remember. And calculation, all the nobody will going to ask, but formula they will definitely going to ask. Okay, you should remember the formula and what it is. And there are softwares like MapMaker, JoinMap, Gmendel. There are different software, but nobody is using. But MapMaker is. But now ICICI mapping is very good software. We'll show how to use it in coming classes. Then where we will get error? Okay. See, everything is fine. Your genotype, your phenotype, you use software, you get QTLs. But where you will get an error? Segregation distortion. For example, I showed that if any marker is not following Mendelian inheritance, not following Mendelian inheritance, means there will be a segregation distortion. Because it's the main assumption in Mendel's laws. We discussed in detail what is segregation distortion. For example, you should get, for example, male and female. For example, A and A gamete should be 50%, 50%. But due to some lethality, you will not get any of this gamete. Only A is possible. This is one type of segregation distortion. There are several types of segregation distortion. If it is there, then you will get error in your like error in mapping. Then interference, we discuss how interference will affect your mappings by creating double like I have to use Kosambi correction factor to avoid this. Then you should not have missing marker data. Means, for example, you are using 200 polymorphic marker. Some of the marker you will not going to get any bands. Like it will not amplify in the PCR. Then you will going to miss. You should avoid it. While doing PCR, you should be very careful. So that you can get the bands on all the individuals and you will get good scoring. See if this will going to wrong, see here some bands are not visible. 20, 20, 19, 20, 21. This is our missing data. If this missing data will be there, how are you going to map them? That's why you should avoid any missing data and avoid error in genotyping. You have to avoid error in phenotyping. Then size. We already discussed what is BVS effect. Can anyone tell what is BVS effect? Yesterday we discussed. What is BVS effect? Yesterday we discussed, na?
Sir,
Smaller, if we
um,
have smaller population,
if
we'll get sir,
sir,
If
high
if we have
resolution.
a yes,
a
yes, yes. Sir,
smaller
if we
population will
have
get higher resolution.
if we have a population with all combination of recombination, we get a map with a higher resolution.
Yeah, that is the good answer. But if you have a small population, what you will get? Overestimation of the particular QTL effect or a gene effect. Okay? Overestimation. That is only the BVS effect. Okay? We will discuss in detail. Don't worry. But if you got an idea, if you're having a larger population, you'll get all the recombinants individuals. If there you get all the recombination individuals, your resolution of the map will be very, very good. But if the population size is very, very smaller, the probability of getting QTL is less. Even though you get QTL, the effect will be more, overestimation, than the actual effect. This is what the errors you will get in the QTL, like mapping or any mapping, not only the QTLs. Okay, this is what, and the next, the another approach is called as bulk segregant analysis. Okay. Bulk segregant analysis. What is bulk segregant analysis? Anyone have an idea about it? Can anyone have an idea about it? What is bulk segregant analysis?
Just to population manipulation, sir.
Nobody have an idea. From UG, we are reading the bulk segregant analysis. What is bulk segregant analysis? My at least slide is visible now.
Yes, sir.
The definition itself in the slide, na? You at least by reading you can say. What is bulk segregant analysis?
So we use it to identify markers or particles.
It's a rapid procedure of identifying the marker in a specific region on the genome. It's developed by R.W. Michael Moore. Many times, like maybe my classes may somewhat like analytical and practical. Maybe you feel that it's unuseful, like it's not useful. But whenever you're facing any interview in SRF, this is the basic questions we're going to ask. For example, you have a project of mapping for some quality trait or in rice or any, like so many projects will come now. Like if you want to give an SRF interview, RA interview, basic question they are going to map. How rapidly you can map the gene. Today we're going to discuss. In 1991 only, see in 1991 only, means it's nearly 40-50 years ago only, this approach came, the very rapid approach of mapping particular gene. See, you have a 200 individuals, 200 mapping population. You have 1000 markers. You had to genotype and phenotype of all the 200, then you will get the map. You know how much difficulties. Okay. The genotype, the 200 individuals with the 1000 SSR markers. You know how much time it will take. See, see for one marker, this is my 490 RM490 marker. I have actually 177 F2 derived F3 population. I showed you paper and we discussed. Na? I have 177 uh, markers, 177 mapping population, and I have nearly 150 markers. If I want to do 150 markers in 177 individuals, see at one At one gel. This is only one gel. One gel max to max you can able to genotype only 40 individuals or 42 individuals. Means you have to do for one marker how many PCR? How many PCR you have to do for one marker? Can anybody guess? These are the questions they are going to ask in interview. For one marker, I have 177 mapping population, but at a single time, see, for one marker, how many PCR I have to keep for one marker? If I want to genotype 177 individuals, how many PCR I have to keep?
171 PCR, sir.
Means one PCR plate having only 96 wells. 96 wells. Okay.
So you have to
And
go
3
times.
and 6 wells for parents. Means only 90 individuals. Means at least 2 plates you need to keep. For one marker, 2 plates of PCR. For 150 marker, 300 PCR plate. Okay. If you want to run the gel, see this maybe in this gel electrophoresis, I can enable to run only 40. Okay, max to max 40. Okay, and sometimes 80 also possible. Means for 40 means in 90 means 3 gels. 3 gels per plate. 300 PCRs for 3 gels means 900 and gels you have to run. Max to max, one person, if you are very good also, you can be able to run only 6 gels. How many days it will take? It will take 3 to 4 months. Okay, if you are running 6 gels per hour, it is impossible sometimes. See, for running only 150 markers for 177, it will take that much time. This is if you have a 500 mapping population and 500 markers, you can imagine how much time you have to give. It's a very difficult task. That's why Michael Moore came up with one very good approach called as bulked segregant analysis. Means you can cross the extreme individuals, you develop an F2 population or any real population. For example, you take 100 population. Normally, what you have to do, you have to genotype 100 population and you have to phenotype 100 population. But he came up with one approach, you need not to go genotyping of all 100 population. Instead, you can create extreme bulks, two extreme bulks. For example, you having 100 F2, out of 100 F2, 10 extreme resistance and 10 extreme susceptible. You take 10 extreme resistance and 10 extreme susceptible from 100 F2. You just genotype them only. See here, then take 10 individuals, then you bulk into one and bulk all the DNA sample from 10 extreme to another bulk. This is called as resistant bulk. This is called as susceptible bulk. Then you have to genotype only with two parents and two bulks. Instead of 200 and genotype plus two parent, 202 genotyping, you are reducing to only 4 individuals. Means 2 bulks, 2 parents. Means how much effort you are going to reduce. Then how we can proceed? For example, first you have to run, for example, you have to take 1000 markers. You are running between the two parents. You are getting nearly polymorphic markers like this. See, A, B, C, D marker are polymorphic. Here 4 markers are polymorphic between the parents. And use this 4 polymorphic marker between the bulks. You run in F1 also, nothing will happen. Between the bulks. See, these are resistant bulks. And here in parents, 4 are polymorphic. But in bulk, how many are polymorphic? This is bulk 1 and bulk 2. How many are polymorphic?
With two, sir. One is polymorphic.
Only one is polymorphic. And this one polymorphic marker is linked marker. means the marker which is polymorphic between the parents and also polymorphic between the bulks. That marker is your linked marker. Okay. This is what bulk segregant analysis means. The you are using a segregating population. From segregating population, you have to bulk the extremes. You have to bulk the extremes from segregating population and do your molecular marker analysis. Then you do mapping. This is a simple procedure developed by Michael Moore et al. Now you got what is bulk segregant analysis? Any
One
doubts
doubt,
regarding
sir.
it?
Sir, one
Yeah,
doubt,
yeah,
sir.
yeah,
So,
of course.
so we are here phenotyping only and choosing for extreme phenotypes, only for
No, 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 no.
that purpose.
See, if you want to select the extreme phenotype, you have to phenotype all the
Yes, sir.
individuals.
Okay.
See, I am telling after phenotyping only you can able to select extremes. Na? Without phenotyping, how you get to know which
Yes,
is extreme, which is not extreme?
yes, sir.
It's a
I
simple
am
logic.
asking only at that case only we are going to phenotyping, no other phenotyping anywhere, isn't it, sir?
What? I'm not getting what is your doubt.
Sir,
Can you repeat your doubt?
just yes, sir. Only one time we are going to phenotype, sir. In all
Yeah, one
those,
time only. One time only.
yes.
Here you will not going to do phenotyping anywhere. You just run the marker. What is your doubt? I'm not getting. One time only. First you phenotype
No, sir.
all
No,
100
no.
F2. Then you
My,
create extreme.
I got confused. Sir. No, many major doubts.
Okay,
Only that mistake.
okay. Then you phenotype all 100 F2. Then select extreme 10 or extreme. It depends again how to select. It's again a very statistical measures. Why only 10? Like it's 10% of your total population. If you're using 1000, what is the 10%? If you're using 1 lakh, what is the 10%? Like this, you have to bulk the 10% of the individuals. There is a statistical measure. Again, I'm not going in detail how it will work. But you select the extremes, then you create the two bulks, then run marker between the parents. Instead of running the polymorphic marker in 100 individuals, run only between the bulks. The marker is polymorphic between the bulks and between the parents, that is your linked marker. This is what a bulk segregant analysis. Okay, understood what is bulk segregant analysis? And in exam max to max, they will going to ask what is bulk segregant analysis, who developed it, and when it is developed. And it's a rapid method. See how rapid it was. Just four individuals, like two bulks, 2 months. You just run. I think within 1 day you can able to do your cover and identify the linked mark. If you have the phenotypic data of 100 individuals, within 1 day you can able to identify a linked marker. That much rapid. That's why it is mostly used nowadays. What are the things where QTL seek or BSR seek, selective genotyping, selective GWAS, all the things are based on the core principles of bulk segregant analysis. If you are interested, please watch my video on QTL-seq and BSR-seq in YouTube. This is my first video. Please watch it. Okay. Then, what is the principle? I told the marker between the parents and the marker between the polymorphic marker between the parents, polymorphic marker between the bulks, that is linked mark. Why it is so? Please read it. Okay. It is based on the principles of NILS, near isogenic lines. Means the bulks are look like artificial nils. Bulks are artificial nils. Why it is artificial nils? Because actually it is again a different, very, I think, very logical thing. What is nils we discussed? What is nils? Near isogenic lines. Hello? And there are
Differ by some genes only, sir. 2 or 3 genes.
the rest all the genome is same. Means the two individuals The genome is completely same, but they differ with only 2 to 3 genes. Means that's why they are near isogenic. Your bulks also, they are artificial nils. Artificial nils means between the markers, between the bulks, what all the polymorphic marker you are going to get that because we are creating the bulks based on the phenotype. Means we are doing extreme phenotyping. Means the alleles which are responsible for the particular resistance or for particular susceptible only pooled in the bulk one, and in this allele responsible for the susceptible only having maximum frequency in the bulk 2 bulk 2 susceptible bulk. Means they differ only with the trait of interest. Means here resistance or susceptible. Maximum alleles here is resistance, and in another bulk maximum alleles frequency is susceptible. But rest of the genome, rest of the background genome other than the resistance are random. You can't say it as same, but you can say as random. Means in the both the case they are random. But when you look at your trait of interest, that allele frequency is maximum here, and that is not there in this another. Means the if you get the difference in the marker, means they are associated with this difference in the frequency of the resistant allele and susceptible allele. But rest all other genome is random, like nils. Nils is all other background genome is same, but between the bulks, the background genome is completely random, but they are only differ with your trait of interest. That is actually the logical idea behind the bulk segregant analysis. I hope you not got this point. You should have a brief knowledge of population genetics. What is the frequency when you do extreme phenotyping? How the enrichment of the frequency of the particular alleles in these bulks. You should have that knowledge, then only you're going to get. But if anybody asks you what principle the BSA works, means it based on the principles of near isogenic lines. You should at least able to answer it based on the principle of near isogenic lines. Okay, understood what is bulk segregant analysis and what is the principle of bulk segregant analysis? Okay,
Yes, sir.
this is we discussed already. Okay, then tomorrow I think I will going to discuss what is QTL mapping. I have so many slides. I think how to develop the QTL mapping, how to use the threshold values. Okay, then after that I will create association mapping. I have everything. No need to worry. I will just having different models of association mapping, and we'll discuss some questions also and different marker schemes, genomic selections, case study some of these. Then future like we'll discuss some questions also, previous year questions. I think I have questions on markers. I'm searching that.
Sir, slides are so good, sir.
Thank you, thank you. No, I'm searching the questions, question bank of markers actually. Actually, after every topic, I used to discuss questions like this. For example, after genome editing, uh, I used to discuss the questions like this. We'll discuss actually. For marker class, I made, but it is in separate PPTs. Okay, I think I will. This I will not include it here. But don't. I will take regular classes because I have everything already. But I have like nearly see this is nearly a 700 slides. Okay. Okay. Any doubts up to here about tagging, gene tagging? How to use nils for tagging? What is bulk segregant analysis? You have this. What is marker systems? What is mapping populations? Basic idea of gene tagging. Then only we're going to get QTL mapping, association mapping, QTL seq, RNA seq, all those things. Please read everything because from tomorrow's class is very much complex. Having like in QTL mapping, you have to use single marker analysis, you have to use interval mapping, compositional mapping, Bayesian approach of mapping. You should have very sound knowledge of the statistics, frequencies like Hadwinsburg laws, and what is structure of the population. You should know everything. Okay, please read. Try to read them. Tomorrow we'll start. Okay. Okay. Bye. If you have any doubts, you can ask. Abhishek, Mandeep. No doubts to hear.
No, sir.
No, sir.
Thank you. Thank you, Varun. Bye.
You, sir.
Good
Thank you,
night.
sir.
Good night. Good night.